All right, let's get started on today's class. We have a quick announcement. So this year we have a lot more students registered for the course. By this time we have a total of 90, which is about 20 more or 25 more than last year. As a result, the lab, the previously registered lab location is not big enough, at least on the Cambridge side. And so we have changed the lab location into Science Center 309. We are trying to find a bigger room to host for a little longer time. The current lab time is only an hour, 15 minutes. We will see whether there's another group coming afterwards and whether we have to move to, say, the Science Center first floor, that food, resting area. But for now, this is the room we can reserve. We'll try to resolve this. Actually, there are 2 days that's different. Today is one. Today you are gonna be in Science Center Hall A. Okay, and then another date is April 9th. And so you will have a new lab location. All right, so now let's continue with lecture. We talked about RNA-seq in the last lecture, and today we're gonna talk about differential gene expression. We mentioned that the sequencing read distribution actually does not follow a normal distribution. Therefore, the standard tests that people are very familiar with, such as t-tests, would not really work in high-throughput sequencing, at least for RNA-seq. However, there are some simple transformation you can do. In fact, when RNA-seq was first available, there are many groups trying this. They look at the data and they see what kind of distribution it follows. And the very simple thing is, sometimes when the data does not follow normal distribution, you can try to take a log, and after the take the log, they see, oh, okay, actually, if you use the log of normalized read count on genes, and they actually follow a normal distribution. And so a question for the audiences here is, how do we know whether something follow a normal distribution or not? So for that, you can test normality by QQ plot. There's a standard R function. Basically, you know that regular normal distribution, how much data needs to be in certain quantile, how much data needs to be in certain quantile, roughly what range, right? And then if you look at the correct or the observed quantile or the observed data range or distribution versus the quantile, if it agrees with the theoretical quantile on this line, that's an indication that you do have a normal distribution. However, if the raw data looks like this, then you know you shouldn't use a normal distribution test. Okay, and so it's been seen that with high throughput sequencing data, if you look at the read count that lands on a gene, and you look at many samples and look at their differences or variations, you can see that after taking the log of the read count, it roughly follows a normal distribution. Therefore, in the early days, there are algorithms that are developed previously for microarrays, because actually for microarray, after we get the gene expression level, it does follow a normal distribution. And so there are algorithms developed such as LIMA, which is really using the linear model to study differential gene expression. You can also now use it on RNA-seq. And so in LIMA, the model is like this. You believe the expression of a particular gene J in condition I in the sample K is equal to the mu. So basically, here every gene has a separate ID, and here is gene I. K is a specific sample, and the mu is the mean expression of this gene over all the samples. You can imagine after you get the expression level quantification, you take the log, you get a matrix. The rows are usually the different genes, and the columns are the different samples. And so you can look at each gene and you calculate the mean of this gene over all the samples. So that's kind of roughly how much this gene is expressed. And alpha is the deviation of the mean from this. This is a particular condition. Okay, so I is say a drug treatment, a disease condition or whatever. And so you assume that there is potential deviation of some treatment on effect on this gene, and that would deviate from the overall mean. But if you look at all the different conditions together, the sum of that difference should sum up to 0. That's why you have the mean, right? All the different samples are kind of normalized. Then when we are trying to compare the samples in condition 1 with the samples in condition 2 on this particular gene, what we are really trying to ask is this conditional effect on gene J compared to the control on this gene J, is that effect equal to 0? Is it equal to 0, this is what we call null hypothesis. We don't reject the null hypothesis. We'll say this gene between the treatment and control are not differentially expressed. But if you believe that they are different, then this will not be 0, and you will reject null hypothesis and call this gene as differential. And since if you take a log of the read count, the log, it follows a log normal. You can use a standard t-test or some modified t-test to do this. And the Lima algorithm actually does this. You can use this, basically, you provide a gene expression matrix, which has the samples in columns and the genes in rows. And then you also need to specify the design matrix, which says, for example, in this case, the first 3 samples belong to a treatment 1 condition, the next 3 samples belong to a treatment 2 condition, and the last 3 samples belong to the control condition. And then you also need to have a contrast matrix, which specifies which type of comparisons you want to do. For example, the first type of comparison I want is to look at the treatment 1 compared to a control. And in this case, the treatment 1 will be 1, the control will be -1. And so the algorithm will already know that you're going to use these 3 samples to compare with these 3 controls. And then if you want to say, okay, I want to do another comparison, which is a treatment number 2 compare with the control sample, then treatment number 2 will be 1, the control will be -1. And so basically in this contrast matrix, you can specify what type of comparisons you are interested in getting. And so basically, the algorithm will use this type of generalized linear model to estimate across all the samples, what is the mean, what is the effect on each treatment, and whether there is a significant difference between one condition and another condition. And so this works to a certain degree, except that log normal is kind of an overcorrection of your data distribution. And so if you do log normal, yes, it will work to a certain degree, but it's less sensitive in detecting the truly differential genes because you're shrinking this data distribution too much. And so later on, a more sensitive method is modeling the sequence read count distribution by negative binomial distribution. We briefly discussed this. Negative binomial distribution is different from Poisson distribution in that a standard Poisson distribution, the mean and the variance is the same. And so the black line would indicate what that would look like, which is, as we mentioned, for example, you can ask within an interval, if it's completely random, what's the chance that if there's a small probability, there's an event that's going to happen in every small interval. You're asking if we look at a bigger interval. So supposedly you say the probability that the ER will see one patient in 1 hour is a tiny probability. What's the chance the ER will see X number of patients in 3 hours or 5 hours? So this is the same. You are asking if the probability of you seeing one read in every 100 base pair, it's a small probability because the genome is huge and you're looking at one read that lands in that 100 base pair. It's a small probability. Then you ask if my transcript is say 1 KB long, what is the likelihood something lands there? For Poisson distribution, the mean and the variance would be the same, but what people observe that is in real sequencing, there are different type of other biases. And so the end result is that the variations usually is bigger than the mean. And so the negative binomial distribution really capture this. So the strict definition of negative binomial distribution is the number of success before the first R failure if the probability of success is P. And then if you really in this interview, you observed X number of success, what is this probability? There are some standard equations you can get to this. And this is an example of some of these negative binomial distributions with different R and Ps. And you can see here, this data spread is quite different from normal, especially, this kind of look like normal, except that the spread is much bigger. This is an indication of this over dispersion, okay? And so question is, how do we really capture this over dispersion? In any type of, I think most of the time when we are dealing with gene expression, modeling variance could be tricky because in day-to-day gene expression experiment, very often you just treat one drug, you get 2 to 3 replicate, you are trying to estimate the variance in the data. This could be tricky. Normally if you don't have 25 or 100 numbers to estimate the variance, you could be quite off. And so in microarray or RNA-seq experiment, sometimes you only have 2 to 3 replicates in each condition. So how do you really look at the variance? So we use this example to demonstrate. In this case, we don't have even a single don't even have 2 replicates. It's a single replicate. Supposedly you have condition 1 replicate, and condition 2 in only 1 replicate. You just look at some, say, read count, normalized read count. You can see, say, a pair of experiment or one experiment, you might get data like this. In another experiment, you might get something like this. It would mean from here that the second experiment, whatever treatment you have, is perturbing the original condition much less, right? And so even if you have a single replicate and we are looking at supposedly a gene with this level of differential expression between the 2 conditions, even though we don't have replicate, I think you can already see that this gene in the first experiment, you probably don't have very high confidence that it is really differentially expressed, right? Because the noise in that first experiment is pretty high. Whereas this gene in the second experiment, you are much more confident that it is a differentially expressed gene. And so this demonstrates the power of looking at many other genes to estimate the rough noise in the data, even if you don't have replicate. And so that's what people are trying to do when you look at RNA-seq experiments with 2 to 3 replicate, is by trying to look at the noise in all of the genes together. Okay, so how do you do this? We mentioned that, so here you can see each dot is 1 gene, and the x-axis measures the expression level of the gene, and the y-axis measures the variance of this gene. And you can see here, it follows this, the noise is higher than the variance is higher than the mean. And so you can imagine, normally we try to model this, if we try to model this as a negative binomial, this Kij is the, sorry, maybe the Ij is slightly different from the previous slide. So this is the raw count for gene I in sample J. I think previously we have I as the sample and J as the gene, but you get this idea. So this is the actual raw count that we see for a sample. It follows a normal distribution. There is a small scaling factor. This scaling factor is dependent on the whole sequencing depth of your library, and also the gene length. As we mentioned, if the gene is very, very long, then you have more reads. But if the gene is short, you have less reads. It may also correct a little bit of the GC content on this gene. And this is the actual expression level you want to really measure, right? roughly like a TPM level. You are trying to see how much this gene is expressed, and then this is the dispersion. So you can imagine we are observing the raw count, and we say, okay, it's the reason we're seeing this much raw count is because we are sequencing this sample at these steps, the gene is of this length, and if this gene is expressed at this level and there's this much noise, that's how much read we're gonna see. And if we were to draw this figure, you will see something like this, and we can actually model this, the theoretical variance of every gene by this equation, which is the mean ij plus the alpha mu squared. So if we don't have the second term, we are saying that the variance is equal to the mu. That's a regular Poisson distribution. But with this additional term, we are trying to capture this over dispersion. And you can see here, if the sample average or mean is higher, then the dispersion is also a little higher, which kind of captured the situation here, okay? So you can see here the dispersion for gene I is associated with its only expression level, because if the expression is here, the variance would be lower, but if the expression is high, the variance will also be higher. But the theoretical variance, which is kind of this blue curve, we estimate this by borrowing information from all the other genes to really get this curve. And so how would that work is, in algorithms such as Lima and DESeq2, the actual variance you observe from very limited sample, these are written as these dots outside the line. The red curve in here is similar to the blue curve in here, which is to really capture how much is that alpha value. You know, how much is the correction are you trying to how much are you trying to correct? Basically, when the data is this is the dispersion divided by the mean count. You can see basically when the sample is at the read count is very, very low, the standard error would also be higher. But when the expression level is higher, the standard error would be lower. And so with every gene, you do the estimate, you try to correct it a little bit towards that theoretical line. So here is the red, yeah, you can think about either the red curve in here, sorry, in here, or the blue curve in here. Okay, so we are trying to correct the variance by looking at the observed variance for every gene, but also the theoretical variance for a gene with that roughly that level of gene expression. And so if the variance is too high from that gene, you try to shrink it a little bit. But if the variance is too low, you try to kind of enlarge it a little bit. The level of correction depends on a few different things. One is the actual expression level, because you can see here, depending on the expression level, your correction is different. And also, how much is this gene varying between the different samples in this experiment? And third is, how many replicates do you really have to estimate this variance? Yeah, question?
How does this work if you so like I know like different contingency analysis, sometimes you might want to take a function of the read count, right? So how would it work if you reparameterize whatever?
So in this situation, the DESeq2 will just, so basically you look at the different algorithms that use different, you know, they're actually, I would say for this course, over the years, we have tried many different algorithms for differential expression. There's DESeq2, there's EdgeR, people use Lima, they use VUM, and they all have slightly different parameterization in here. And at the end, it's determined that you just use kind of a normalized read count. This normalization is not trying to normalize between a sample that's sequenced at 3 million read versus 300 million reads. That's too much. You only normalize a little bit between, say, 52 million reads versus 55 million reads, just a little bit, right? And then you just look at that normalized read count, then you can estimate this, okay? Yeah, so basically, the red curve is kind of the correction of the variation or the theoretical variance of the genes at a different expression level. And each of the dots is the observed variance that you see at each gene, and you correct it a little bit by still looking at the data you get from each gene, but also looking at all the other genes to correct it a little bit. So to create a more stable variance estimate. Okay, so with that information, for example, if you have DESeq for differential expression, you have sample 1 and sample 2, after we shrink the variance, we can then estimate this probability, you know, whether this is using the original negative binomial distribution, and you're asking whether a particular condition, if you know the mean and the variations, what are variance, whether the observed value is significantly different from the mean. Okay, and so DESeq will give you this calculation in terms of after you find the differential expression, how do you visualize it? There are two different ways people look at it. If you present either of these results to in a paper, usually the reviewer will be okay with that. The one way is called a volcano plot. It's kind of like a volcano shooting off lava. On the x-axis is the log fold change. You're asking between the two conditions. Fold change is just, you know, how many reads you see in the first, how many reads you see in the second condition, and you take a log of that. And the y-axis is a p-value that's calculated based on the previous negative binomial distribution. And obviously, the most significant gene are going to be this blue dot on the left upper corner versus the upregulated genes will be the right upper corner genes. Okay, that's one way of looking at this. Probably a more informative level of differential expression is by looking at something called the MA plot. So in the MA plot, the M is the log ratio or log fold change between the two conditions, and that's on the y-axis. This is similar to the X on the volcano plot. You can see that's kind of similar. Whereas the x-axis in here is the mean or the average expression level of the two, taken at very often taken at a log, so it's kind of a geometric mean. And so every dot is one gene. And so you are asking if the x-axis is the average expression and the y-axis is the log fold change, you are trying to ask if we look at genes at the different expression levels, which genes are different. And the red dots are all of those ones that are considered to be significantly different. So this way you can at least, you can also figure out, you know, roughly how much is the noise and how many genes are you calling as different and how confident you are about those differential genes. And so to give you an example, if we were to just directly use the mean and the variance that we observed on each gene to calculate the differential expression, on the left figure, this is what we get. You can see here, the x-axis is kind of each dot give you a rough idea how much this gene is expressed in general, and the y-axis give you kind of a log fold change. You see that there are a lot of genes that are called as differential expression with huge fold change. This is just because when the numbers are small, you can easily get a big fold change, right? But then you are not that confident about those genes, and it's better not to really call them. These genes actually probably are truly differentially expressed. And so with the DESeq way of normalizing or stabilizing the variance based on this theoretical variance, by borrowing information from all the other genes, you try to, you know, if any of the variants are too far off, you try to move it towards the center a little bit. You can see here, then the low count genes really, you shrink their variance or you stabilize their variance, then these genes are really not called as differentially expressed in terms of fold change, whereas the higher expressed genes will look quite similar from before. This would mean that you will be able to really know which genes are truly different and which genes are noise. Whereas from here, you can see if you do some cutoff, you will be calling a lot of fake differentially expressed genes. Okay, so this variance stabilization is a very good way to increase your signal to noise, mostly to decrease the noise so that you will be able to have better sensitivity to call differential expression. Okay, questions? All right. So the next question is multiple hypothesis testing. So this really is relevant to the question. We have a high throughput experiment, and in the whole genome, there are about 20,000 genes, and we did this differential expression analysis. We tested the expression of every gene between one group of samples and another group of samples, and we are asking, is it different? And at the end, you can rank the genes by their level of differential expression, but where do you cut the line to say, between the two conditions, I have 200 genes that are different, or 500 genes that are different, or 1,000 genes that are different? Can we use p-value for something like this? Because basically, if you use the negative binomial distribution, or if you convert the original data into a log normal, you can also use t-distribution to calculate the differential expression. Each can give you a p-value, right? Can you use the p-value to give you, to decide how many genes to cut?
No, like, suppose you like a piece of 5%, fine, like you have a 10,000 genes, 500 of them will be significant because they are false.
Right. Yeah. That's definitely the right answer. So we mentioned here that when we try to test the differential expression for every gene with a p-value, supposedly on every gene, you decide you want to use 0.01 as cutoff. And if this gene is, if you decide this is not differentially expressed, that's the null hypothesis. If the genes are really differentially expressed, it's called the alternative hypothesis. You will reject the null and really call something to be differentially expressed. If something meets the p-value cutoff, the situation as just the student mentioned, in the genome, we have 20,000 genes. If you use this 0.01 as cutoff, a p-value as cutoff, there are going to be 200 genes that are being that will be called, because you have so many genes. By chance, you will have 200 genes that have a p-value less than 0.01, and so even if you have no genes differentially expressed, you will still call about 200 genes, and probably all of them are wrong. This is similar to a situation like this. Today in the classroom, I'm trying to ask, how do students decide whether they are going to sit in the first half of the classroom or the second half of the classroom? I could try different things. I say, okay, let's first try undergraduates all sit in the front. No. How about people with glasses all sit in the front? No. And all male students sit in the front? No. People who live within or who are from the US will sit in the front, or who wear sweaters will sit in the front. Like if you try enough times, maybe we will see that, oh, all of those who have a sibling and ate noodles yesterday sit in the front, right? We just don't know. But if you would try 20,000 different things, maybe one of them will be a hit, right? Or some of those will indeed be able to separate the students in the front versus the students in the back. But that's just because I've tried many different options. It's the same situation with looking at 20,000 genes and asking whether they are different. And so we need to make sure we control for multiple hypothesis testing. And one way to do this is to make sure that I don't wrongly call even a single gene incorrectly. And this is called the family-wise error rate. So this is the probability of rejecting at most one hypothesis at less than a certain cutoff, which means the probability of not even calling one of them wrong is going to be 1 minus this number. So supposedly, if I want to correct for the family-wise error rate at 0.05 would mean that for whatever number of genes I call, even one of them is wrong, is lower than 0.5 probability, then I have to use on each individual gene a much more stringent p-value. Okay, so the way to do this, we wouldn't go to the statistical deduction, is to use this Bonferroni correction to really control the family-wise error rate. If you have M hypothesis to test, and you want to control the final, whatever number of genes you call, I want not even a single gene to be wrongly called at this level of confidence, say 0.05 confidence, then for each individual gene, our p-value cutoff need to be alpha divided by this M, which means if they say in a RNA-seq experiment, if the alpha is 0.05 and we have 20,000 genes to make the prediction, the p-value cutoff would be 0.05 divided by 20,000, which is a very small number. And so sometimes when you don't have so many replicates, or the data is fairly noisy, you may not be able to see any genes to be differentially expressed, at least at this cutoff. And so this is too conservative in terms of multiple hypothesis testing correction. And so what have been used are this other concept called false discovery rate. And so if we look at the a study like this, so the rows are the truth, okay? The truth is H0 is your null hypothesis. You say the two conditions or the two groups of samples are actually similar. There's no difference in between the gene expression. In the alternative hypothesis, you are saying that actually the two genes are different. And then if you look at the columns here, that means this indicates for whatever statistical test, you are going to really make a call to say they are different, or make a call to say they are not different, which means whether you reject or not reject the null hypothesis. And so what is U in this case? So U is this group, so for a particular gene, the two groups are quite similar, and you also did not call it as different. What is this? It's a true negative, right? This is a true negative, that's U. What is V? It's a false positive, right? Because these two groups are similar, but you call them as different. That's a false positive. T is a false negative, right? These two groups are different, but you didn't call it. That's a false negative. And then the S is a true positive, right? And so, yeah, as we mentioned, the false positives are what we call type 1 errors in statistics. And the T, which is the false negatives, are the type 2 errors. False discovery rate is the V divided by R. R is basically all the things we called. So we are trying to ask, out of all the calls we make, how many are false positives, or what percentage are false positives? That's the false discovery rate. Okay, so basically when you do a RNA-seq experiment, you want to see some genes are different. And even if there are some mistakes in occasional genes, you can tolerate that. Supposedly, if you say, I want to call whatever number of genes to be different, as long as the total wrong calls are no more than 2%, that's okay, right? If I call 200 genes and 2% is false, that's 40 genes, that's okay. So 160 genes are still correct. that's false, controlling false discovery rate at 2%. Okay, and so you can see family-wise error rate is to make sure that out of those 200 genes, not even single one of them are wrong, or at this probability. Whereas FDR just says out of the 200 genes or whatever number of genes you call, X percentage could be wrong. You don't necessarily know which ones are wrong. You just know roughly X percentage of them could be wrong. And so, so the way to calculate this, I stole this from the StatQuest by Joshua Starmer. By the way, in the course schedule, you will see the lecture slides and the videos, and the bottom are other videos I found on YouTube. I would say sometimes, and probably most of the times, some of these other videos are really quite good, and you should watch those as well. I especially like this StatQuest by Joshua Starmer. And so I just did a screen dump from his YouTube video to show you. So this is also part of this homework one. You can see what we ask you to try this. You know, you use a random number generator from say uniform or some same distribution, and you just generate some group of samples to look at their difference. Let's just say. Supposedly you have a random number generator to generate a differential, to generate numbers. Let's just make them simple. If the distribution that you generate this number is a normal distribution, and say you randomly generate 10 numbers in group 1 and randomly generate 10 numbers in group 2, and you calculate their differential expression using a t-test. That will give you a kind of you know, each will give you a normal distribution. And when you look at their difference, most of the time you will not see a significant p-value, but then occasionally you will see a small p-values. But if you look at all the different p-value distribution, it should be uniform. That's what p-value means, because it's the same distribution. You just randomly generate numbers, you test whether they're different. It should be a uniform distribution in here, right? So from 0 to 1, and every data has the same probability of hitting a particular p-value. And this is all noise. But if indeed there are some genes on this experiment that are different because of the treatment, it would mean that underlying there are two different distributions. Supposedly one is a normal, the other is a drug treatment, or one is normal, the other is a disease state. Then when you are randomly generating numbers from the first distribution versus the second distribution, and then test them and look at the p-value, you probably see something like this, right? The p-value is much more skewed towards a smaller p-value because indeed they are coming from different distributions, right? So the reality of the data we are getting is a mixture of the two, because you are dealing with 20,000 genes on this, in the genome, and most of the genes in the experimental condition. I think we can make some safe assumption that anytime you treat with a real cell, live cell with a drug or something, most of the genes are going to be staying roughly the same. The genes that are changing is only a subset. And so most of the genes will give you this type of noise, uniform distribution, but only a subset of the genes, a tiny portion of those genes will give you a true signal that's skewed towards the left. But when you mix them together, this is what you get. If you take a look at a particular experiment, in fact, if you want to test, anytime you test a lot of hypotheses, you might want to draw the p-value distribution to see whether your statistical test is good. Because if you normalize your data well and you've done your statistical test well, you should see a p-value like this, okay? What happens if after you finish an experiment and you run the statistical test and then look at the p-value of all the genes in an RNA-seq experiment, you only see this? What does that mean? If you see something like this in the p-value distribution, it would mean that your data is too noisy. There is no differentially expressed genes that are significant from your experiment, okay? Whereas very often we see something like this. By the way, if there's no differentially expressed genes, the uniform distribution should be, the y-axis should be at 1, and it should be quite even to cover this area. But if there are real signals that some genes are truly differentially expressed, you have more area on the small side, and that's going to take the area away from the rest. And so this area may not really, sorry, these bars may not really reach 1. It might be 0.9 or 0.95 or whatever, right? So it's a slightly lower number than 1. Remember, we're saying that this is kind of a addition, like the sum between the noise and the signal. Therefore, if we say we want to do a p-value cutoff at this first bar, how much is the signal and how much is the noise? We would say actually any p-value, roughly, the noise will already give you this level of p-value below that bar, right? Just random chance, even if the two distribution have no difference. So these numbers or these genes are gonna be the random noise, whereas anything above this dotted line, these are the real signals. And so what is the FDR if you cut off at this level? So FDR is the false positives divided by all the things you call. It would mean that if you were to cut off at, if you are going to call differential genes at this particular p-value, your false positive is, how many genes are you going to call? If you're going to call a p-value at this cutoff, you're going to call everything to the left of this as differentially expressed, right? But out of all of these genes, anything below this dotted line, these are all random garbage from the noise. But everything above it, that's the real signal. So the FDR would be this little area divided by the whole bar. And so then if you were to cut at another p-value, let's say let's cut off at here, it would be, the noise would be everything below this dotted line here, divided by all the genes that are called below this p-value, which includes this bar together, right? So basically the intuition is that every particular p-value cutoff, you know that the A part is coming from random noise, and the B part is the real signal, and the false discovery rate is roughly A divided by A plus B. So A plus B is all the genes you're calling below that p-value. And you know that the noise in that, the potential false positives are estimated based on this lower dotted line, which is kind of, you estimate based on the right side. So that's why if you design a good statistical test, if you capture the correct distribution of your data and you do the right statistical test, after you finish testing all of your genes, that p-value should give you something so that the right side is pretty flat. And you can use that flat level, like extrapolate the flat level to the left to estimate roughly how much is your noise. And the remaining ones you called are the real signals. Okay, and so as we mentioned, in this situation, you just know roughly at each p-value cutoff, how many genes you're gonna call, and roughly out of those, how many are false positives. You don't know which one is the false positive. You just know a percentage of them are gonna be fake. Okay, that's FDR. There is a one-to-one correspondence. Basically, you can imagine at every p-value cutoff, you can estimate the A divided by the A plus B. Okay, and so the FDR value is always higher than the p-value. Because this is basically p-value is whatever cutoff you have. But below that, there are always something that are fake. Okay, and so FDR is basically a less conservative way to do multiple hypothesis correction than the family-wise error rate. there are a very widely used method called Benjamini-Hochberg method. It estimates this FDR. You know, the rough idea is like this. You estimate by extrapolation from the right side. You know all of the things on the right are, that give you the level of noise. And this will tell you how much truth you are going to have. You might see FDR. You might see, depending on the algorithm, they say adjusted p-value. And sometimes you might see Q-value. They all mean the same thing. Adjusted p-value is the multiple hypothesis testing adjusted p-value. That's the same as FDR. Or Q-value is all the same. And as we mentioned, p-value and FDR are always monotonic. So every p-value has its corresponding FDR, and it's also bigger than the p-value for the same gene. So at the end of differential expression analysis, for each gene, based on the negative binomial distribution, you will get a p-value. And by looking at all of those p-values together, you will also estimate an FDR. And usually people take that FDR values. And so in order to really report genes, some people take 1% FDR, some people take 5%, some people take 10%. At least when you say I'm reporting FDR, the readers would understand that you have already done multiple hypothesis testing correction, right? And sometimes people, in addition to the FDR, they also filter by fold change. Say the fold change needs to be 1.2 or 1.5 or 2 fold change. But in general, with this FDR estimate, you can really get a sense of the signal to noise of all the hits you're calling. Most of the time, in reality, is people are comfortable with calling between, I would say, 50 genes and 3,000 genes as different. If after all of this, you get 3 genes that are different, you're kind of wondering, okay, maybe my experiment is too noisy, you know, something is wrong. But if you're calling too many genes as different, you might want to use more stringent, like if you see 5,000 genes as different, maybe you want to use a more stringent FDR cutoff, or you want to also add a filter for fold change, okay? One way to, so you can see basically the level of noise you have in the data versus the signal depending on, so supposedly if I have only 300 genes that are differentially expressed, but I'm doing 20,000 random other genes, the noise is 300 out of all the 20,000. But if instead of 20,000 genes, I only look at 5,000 genes, then the noise level would be much, much lower actually, and the signal would be much higher. And so is there a way we can reduce the multiple hypotheses we're testing? If you have a lot of noise, like if you have to test 10,000 genes, well, of course in human we don't have 10,000 genes, but suppose that you have 10,000 noise versus 300 signal, it's going to be a lot harder to call. But if you have only 5,000 genes and 300 signal, it's easier to call. But how do we reduce the number of hypotheses in this case? So algorithms such as DESeq will try to ignore genes if it has too low gene expression level in too few samples. This gene is hopeless, right? What's the point of testing differential expression? You know you will never really actually reach a p-value significance. So with DESeq, there are some simple heuristic to say, oh, if this gene is expressed, say you have 6 or 10 samples, and you only see a single number in one of the sample, and the remaining samples are all 0. It's hopeless. This will never really reach p-value. And so DESeq will use some simple heuristics to remove those genes that have too low expression level in too few number of samples to reduce the total number of genes to test. This way, the number of noise will be reduced and the signal will be higher. Okay, yeah, but at the end, most people are most comfortable with usually a few hundred differentially expressed genes. Questions about FDR? I would, yes.
Would you explain again why FDR is larger than p-value?
Because if you cut off at 5% p-value, And if there is no differentially expressed genes, do you know what your FDR is? It's 100%. All of them are fake, right? But if out of the, supposedly you do a cutoff and 50% are real and 50% are true, then you have 50% FDR, and that p-value you can see here. In each of these are all of your fake ones. And these are your correct ones, right? You are trying to estimate this percent of fake with the total compared to all the others. The bottom ones give you that p-value, right? And so FDR is always bigger than the p-value. Okay. Yeah. So I would say the best thing statisticians have done to teach biologists is about multiple hypothesis testing. With high throughput sequencing, I would say before there is any high throughput experiment, people didn't really care as much about statistics. You know, biologists, they just feel like, oh, if I see different, anything different, I'll report it. I have a paper, that's great. Starting from gene expression microarrays, when people started looking at thousands of genes or tens of thousands of genes, they realized that you need to learn about multiple hypothesis testing. And I think that's really something statistics helped with biologists. And so basically after a microarray experiment, you use DE-seq negative binomial distribution to call the differential gene, then use the FDR to estimate how many genes you really want to call, so that whatever number of genes you call, maybe only 5% of those are wrong, and the remaining ones are going to be correct. Okay? So the next part is about gene ontology. When you have differential gene expression, people are interested in knowing what those genes are, but imagine reporting this in a paper with a list of gene names. As we mentioned, people are comfortable with seeing 300 genes that are different. You know, if you have a paper and you say, after we did this experiment comparing cancer and normal, we see the following 300 genes as different, and you list the whole 300 genes in the paper, it will be kind of very difficult to understand or kind of boring, right? And so people want to say something about those genes. Do they have certain functions? Are they enriched in some pathways or something like that? Yeah, so how do you report the differentially expressed genes or a group of genes, right? Are they enriched in pathways? Are they enriched in some functions? Are those genes or proteins localized in some complex? Are they all in the nucleus? Are they, are they all enriched in something? And so that means you need to give a gene some kind of annotation. The motivation of this really stems from people starting to look at how genes are named. Years ago, when people are just still, we don't have the human genome or whatever species whole genome yet. Every scientist who discovered a new gene are free to name their gene with, they can name the gene themselves. That's kind of an honor. It's like when you find a new star, you can name it whatever you want. And so people who discover new genes can name it. And very often they give it some interesting genes, gene names based on a particular phenotype. They say, this gene is related to a fly having white eye or some strange wings or a wrinkle pea, right? So they can name the gene some crazy names. But then after a while, this name will go a bit crazy in the field because in different species, even in the same species, our genes are named quite differently. And so a consortium of biologists got together to say, we need to really be consistent and try to annotate the genes better, just so that later on we can understand what that gene is doing, especially if you use a model organism like cell lines or organisms such as a fly or a worm or a mouse. You understand that that fly gene is roughly the same as a human gene if you annotate them correctly. Yeah, so the reason we care is that this way you can effectively communicate biological knowledge. You can also organize and summarize these annotations in a better structured way. And the most important thing is you can allow effective and meaningful computation on the gene annotations. So after you got the 300 genes, if each gene is annotated with a code, at the end you can look at all of this code and say, all of these genes are related to certain pathways or certain functions. And so based on this, the Gene Ontology Consortium decided to divide to annotate the genes in the following way. Every gene is given 3 terms. One of the terms is to annotate the molecular function of the gene. What is this gene's job or abilities, right? So you can have transporters or transcription factors. So, you roughly say what this gene does. The second code or annotation for this gene is biological process, which says what kind of event, what kind of pathways does this gene participate in. These are things like cell differentiation, maturation, development, or metabolism or something. There's some pathways in here. And the third annotation for each gene is a cellular component, which describes the location of this gene after it's made into a protein. Because when it's a DNA, it's always in the nucleus, but maybe after the protein is made, they go to a specific subcellular structure or is part of a macromolecular complex. These are things like this protein is in the nucleus, it's on the cell membrane, it's in the mitochondria or in different proteasome and so on. And so every gene is given these 3 terms. But within each term, you can see here, if you go to the Gene Ontology website, they will show you something like this. Initially, you have everything, which have 183,000 different terms. And if you open this, by the way, if you see a little plus, it means if you click on it, it can open up. And so if you click on this, it will open up 3 things. One is the biological process, one is the cellular component, and one is the molecular function. But if we open the biological process, you can see here, it's there are more items in here. For biological process, there's behavior, unknown process, cellular process development, physiological process, regulation of biological process, and viral life cycle. Okay, and if we were to click the viral life cycle, it will go to even more terms, right? So basically, you can annotate. So this is a tree. Initially, in the tree, you have all that's kind of the bulk of the branch, the stem of the tree. And then when it first starts to branch, there are 3 different branches. One is the biological process, one is the cellular component, one is the molecular function. Then the tree has 3 branches, and each branch will continue to branch out, and you will have more and more specific annotation. For example, if we look at a viral life cycle, there is the latent virus infection. There is the viral spread within host. So you can see it really gets much more specific. And each of them, it tell you, so you can see here, it has a number. That's the gene ontology code. That's the number that people or computers use to do computations. And also, these numbers tell you how many terms are underneath this big family of terms. And so as you blow up more and more of those plus signs, the branch you will see more and more smaller branches, and each branch will have fewer terms, and eventually there might be very few. Then see the end of the annotation. And so you might also notice there is this I and also the P. This indicates the relationship between the parent node and the child node on this tree. And the most common relationship is P and I. So the I means something is A. For example, 5' UTR is part of a, sorry, the P means part of. So 5' UTR is part of a transcript. Or you can say in a protein complex, one gene is part of the complex, another gene is part of the complex, third is another part of a complex, right? Whereas that's the part of is a, so for example, mRNA is kind of a transcript, right? It's one type of transcript. tRNA is another kind of transcript. RNA is another kind of transcript. And so the is-are is another relationship that denotes a more specific thing for the general class, right? So for example, cellular morphogenesis during differentiation is a more specific kind of cell development, whereas cell development is part of cell differentiation. Yeah, so basically it denotes, you know, the relationship. Also, actually, the same term could be annotated as multiple branches. Sometimes, say, transcription factor, you can see that, or transcription regulation can sometimes go to both the biological process or the molecular function. So you can see here, if you look at the 3 branches, the biological process have 111,000. Cellular components have, yeah, each of them has about 110,000. And if you were to add these 3 together, you should expect 300,000 different total terms. But then the total term is only 183,000, which means there are some terms that will appear in multiple branches. But the Gene Ontology Consortium made sure that there is no cycles, which means one term is the parent of another, and this one in another part of the tree is the parent of the first, and then you create a cycle. That's not very good. So Gene Ontology Consortium make sure it's a acyclic tree. It's a tree, and you never have the parent and child relation reversed. It's always who is more important and what's more, or which one is more general and which one is more specific. And so with this information, every gene is being annotated as 3 numbers. One is on the biological process, one is on the cellular component, and one is on the molecular function. So for example, if a gene on the biological process is annotated as viral spread within host, it would mean that this gene also belongs to the viral life cycle and also belong to the biological process. So anything that this gene ontology code is, all of its parents are also true, okay? And so depending on how well we know about each gene, if it's a new gene, people don't know anything about it, it might be annotated by some very general terms. But if this gene has been studied by over 100 years, people know it really, really well, then it will be annotated with kind of a branch, very small branch of a very specific function, very specific process. But if it's annotated to have that specific function, all the parents' nodes annotation also belong to this gene as well. Okay, so that's how the gene ontology are assigned. Every gene has 3 numbers, and if the gene is annotated as 1 number, all its parent trunks, those annotations also belong to this gene. Therefore, after we finish, after the Gene Ontology Consortium annotates the gene clusters, we have situations like this, which is we can look at each gene ontology term, some Go term X, which is a number GO 1, 2, 3, 4, 5, 6 is like that code. We ask, okay, in the whole genome, 20,000 genes, how many genes are annotated to have this particular gene ontology code? You say, okay, well, there are about 100 genes that with this gene ontology term. And then you ask, well, in this gene expression RNA-seq experiment between the treatment and control, how many genes are upregulated? supposedly we have 200 genes that are upregulated, and then we ask how many of those upregulated genes have this gene ontology. If it's 80, we can calculate, is this a significant enrichment of genes belonging to this particular gene ontology terms? Okay, so for that, it's a goodness of fit test. And so for that, we use either a chi-square test or Fisher's exact test. For example, we say, in the whole genome, we have 20,000 genes. And in this experiment, 200 genes are upregulated. The remaining genes are not upregulated. But then if we look at how many genes in the genome are annotated with this gene ontology term is 100, but then 80 of them are upregulated, we can calculate the expected number of upregulated genes that carry this gene term. And that's calculated by the total number of upregulated genes, the total number of genes with that gene ontology code multiplied by the total number of up genes divided by the total genes. So 100 times 200 divided by 20,000. By chance, you only expect to see one gene that's upregulated to have this gene ontology term. But what you observed is 80. It's a really, really high enrichment, right? You can also calculate another term. So this 99 is calculated by 100 times, all the remaining not upregulated genes divided by the total gene, that's 99. And this number is, if we look at, it's all the genes without this gene ontology code that are upregulated. These two multiplied divided by the 20K, that's 199. And so each of the numbers in the parentheses is what you expect to get by multiplying the total in the column and the total in that row and divided by the total genes. And so with this, 2x2 table, that's the standard 2x2 table, you can calculate this on a regular chi-square test. This is the observed. The numbers in these four here, that's the observed, subtracted by the expected, which is in the parentheses. And you take the square divided by the expected, and you can check the chi-square table, that will tell you the p-value. But you can probably guess that this is a ridiculously significant p-value, right? And so we can do this. By the way, if this is enriched, what you will report in the paper is, instead of giving the gene names of 80 genes, which is very hard to spell out, you just use that gene ontology term that's associated with these genes. Genes related to viral transmission are enriched in this process, enriched in this experiment. So you can use a text to describe that gene ontology term as enriched in your study, rather than giving it 80 gene names. Okay, that's how you can present the differential expression in a more interesting, biologically interesting way. And so question is, for this, do we need to do multiple hypothesis testing? So remember, we are testing one gene ontology terms. In all the gene ontology, there are 180,000 terms. We need to do this one term at a time, over 180,000 times. So after you calculate the differential expression, you get the 200 differentially expressed genes. You will need to do this test for all the 180,000 gene ontology terms. And each term you ask, do I see this term enriched in my upregulated genes? Therefore, yes, you do need to do multiple hypothesis testing. Okay, and then that's also by FDR. And so if you go to the gene ontology website, there are actually hundreds of tools for a while. I think nowadays people use a few, like such as Amigo, Gorilla, or Great, and also David. For example, David is an online tool. You can paste a number of genes in there that's your differentially expressed genes. Usually, if you have too few genes you put in there, it's not going to give you anything significant. If you have 3 genes, what's the chance that they're going to enrich in anything? If you have a few hundred genes you input there, it will show you, oh, these genes are enriched in some pathways. But in order to get that, it tests every possible gene ontology term to see which one is enriched. But David also have an upper limit. It doesn't want to take more than 3,000 genes as your input. Usually from a differential expression, you wouldn't expect over 3,000 genes to be different, right? And so you can use this approach to figure out whether your list of a few hundred differential genes are enriching something. It will show you, this group of pathways that they all have similar gene ontology pathways. So David automatically group them together, and it will give you a p-value of that enrichment based on the chi-square test, and also it will also give you a FDR, which is the multiple hypothesis corrected p-value of a pathway that's enriched. And then you want to use the FDR adjusted one to decide how to report this in your paper. Okay, so for today, I don't think we will have time to talk about gene site enrichment analysis. So I want to do a very quick summary. Basically, for gene expression, differential expression, if you can take the log of the counts, then you can basically normalize the data. Once it's normal distributed, you can use some linear models, t-test. But you can also just use directly on the counts, and that follows a negative binomial distribution, and you can use DESeq. And for both methods, they try to estimate or stabilize the variance that you measured from very few replicates by borrowing information from all the other genes. And with any type of differential expression, because you're testing all the genes in the genome, we need to make sure we do multiple hypothesis testing correction. And the family-wise error rate or Bonferroni correction is too conservative. So what people use is the false discovery rate. And as we mentioned, usually you want to use something like 5% FDR using the Benjamini-Hochberg approach, right, to estimate the rough number of false positives based on the distribution of all the p-values. And then after you decided I'm going to report these 360 genes as differential, how do you really tell the readers what these genes are? You look at their gene ontology code, right? So each gene has a code for its molecular function, for its biological process, for its cellular component. And so you test every gene ontology code to see whether your differential genes are enriched in your list. And there are tools such as David that can automatically help you do this, but there you also need to make sure you do multiple hypothesis testing correction. All right, great. That's good for now. Okay, let's get started with the lecture. So last time we talked about differential expression, multiple hypothesis testing correction, and then gene ontology analysis. Today, there was one section that we didn't quite finish, which is gene site enrichment analysis. So this was first invented in the microarray days, but later on people also use it for RNA sequencing. So there are some experiments when you compare the two conditions, there might be no or very few genes that are significantly differentially expressed if you correct for multiple hypotheses. But it could be that a group of genes that are related to certain functions are only slightly differentially expressed, but coherently. So for example, supposedly we have some sample in one condition and another condition. If you look at all the differential expression, every dot here is one gene, the X and Y axis is the, say, expression level of the two conditions. You might see there are not many genes differentially expressed. There might be 2 dots here, but they might even be the wrong answer. However, if you look at genes that are annotated to have the same gene ontology, biological function, or they're annotated to be in the same molecular complex, or they have the same pathway, you might see in this case, these genes that are annotated as sharing one particular gene ontology category are colored red. Did you notice something special about these genes? They are only slightly more expressed in the x-axis and less expressed in the y-axis. If you look at one gene at a time, they are definitely not significant. But if you look at all of those genes together, you might notice a little bit that they are a little bit shifted to the lower right half. And so that's how gene set enrichment analysis is initially developed. And to do something like this, in 2003, the original paper by Vamsi Mutha, who is now a faculty at Harvard MGH, he used this Kolmogorov-Smirnov test. This is a non-parametric test. And so after a gene expression, differential expression analysis, instead of selecting a particular cutoff to say, I want to only have X number of genes at differentially expressed at certain fold change cutoff or at certain FDR cutoff. Instead, you can rank all of the genes, all 20,000 genes from the beginning to the end based on their level of fold change. And so in this case, if you look at all the genes in here, I think this dotted line is the genome-wide distribution, which is a cumulative fraction function, which is what fraction of genes have a fold change below this level. So you might see initially there are some genes that have fold change. By the way, this is already a log fold change. Sorry, this is a fold change. So you might ask what fraction of genes are below 0.1 fold change, which means the treatment condition, treatment and control are tenfold different, right? And you might have very few, but then you ask how many genes are differentially expressed with one fold change, which means there is no differential expression. How many genes are differentially expressed below a 10-fold change? So you can see pretty much all the genes have a fold change below 10. This may not really be the best example because in reality, what percentage of genes do you expect to have fold change below 1? Fold change of 1 means there's no differential expression, right? And so you probably would expect half of those genes. So this may not really be the best example. In the real case, the genome-wide distribution should, at fold change of 1, pretty close to 50%. And in this case, neither conditions are like this. But imagine you use all the genes in the genome to generate that background distribution, and then you ask, if I'm only interested in the genes that are annotated to have a specific gene ontology code, and take those genes. For example, I want to look at all the genes related to mitochondria, or I want to take all of those genes that are related to chromatin remodeling, right? So all of those genes annotated with a particular function or complex, right? Then you look at those genes' differential expression and ask what percentage of those genes have a fold change below 0.1, 1, and at every percentage. And then in the process, we ask what is the separation between the genome-wide distribution and the distribution from your gene set. And this D is measured as the maximum difference between the gene of interest, the gene set of interest and your genome-wide distribution. And there is a non-parametric test called the KS test that can give you the p-value of significance. And so this was really quite interesting when people found that after maybe their experiment don't perturb the cell as much, they don't see many differentially expressed genes, but then they can run the gene set enrichment. You just give all the genes a rank order. and then you enter this whole gene set in that order into the this gene set enrichment analysis tool, and then the tool will automatically look at every gene set that shares the same gene ontology code. And then check each gene ontology or Go code whether genes with that Go annotation are significantly different from your genome-wide differential expression level. And so question is, do you need to do multiple hypothesis testing in this case? Yes?
If you run this for multiple specific functions,
yeah,
you're just testing the distribution of a single set of specific functions.
Yes. So the answer is, if you are only interested in a specific gene ontology annotation, then you don't need to do multiple hypothesis testing. Most of the time when people run this analysis, they just provide the differential expression gene order, and they don't a priori know what gene set might be interesting. And so this GSEA tool will automatically test all the gene ontology code. Then you have to do multiple hypothesis testing because it does test 180,000 different Go codes. Okay. Yeah, so the user input is all the genes ranked by their differential expression. There's no cutoff. You don't need to set the cutoff. And then the algorithm will automatically check for genes with specific gene ontology annotation that are involved in the coordinate up or down regulation, right? So you evaluate the significance as a whole. The key is to define the gene set before looking at the data. You can't just say, what if I take all the genes, the top 100 genes that are upregulated in my ranked order list and look at their significance? Of course, those are going to be super significant, but you cannot look at the data first before you do this. And you have to define the gene set ahead of time and also correct for multiple hypothesis testing. Later on, this tool has been gradually improved and is developed by the Broad Institute between Harvard and MIT. And now there's a tool they evaluate. So you can see the idea is like this. If you look at all the genes that are either upregulated or downregulated genome-wide, this is kind of the genome-wide order. And red is the upregulated genes and the blue is the downregulated genes. And every time you can see this is the genome-wide distribution and the level of differential expression, a pretty smooth curve. And the black bar here indicates the genes that are annotated to contain a specific gene ontology code or whatever gene set that this provides. And so you can see here, there are genes that are more skewed towards the left. Therefore, in the gene ontology analysis, you see that there is something called a leading edge subset, which means that the genes that are annotated to have this particular gene ontology, they are more enriched in the upregulated genes in here. So you can see, if you look at the black bar, they are skewed toward the left. And that means that genes annotated as these are overall more upregulated. Whereas this is another situation, you can see this is the genome-wide distribution. And then in the leading edge analysis, you see that genes that are annotated with this particular Go code, they are more skewed toward the right side, which means those genes in general are more downregulated in here, right? So that will give you a kind of a downregulation enrichment. And so this can be used to evaluate enrichment for both upregulation and downregulation. And since then, the Broad Institute has really expanded this into something they call the molecular signature DB. The paper was published in 2005, but since then they have continued to collect the data and put them in the MSigDB. So the idea is, in addition to genes that are annotated to belong to specific gene ontology as one group, you can also look at the previously published studies and look at those genes that are upregulated. So supposedly you take all of the publicly available expression data, including microarray, including RNA-seq. And each of those experiments, when people are perturbing the cell or comparing the differentiation status or drug treatment, disease, or normal status, and then they look at a set of differential expression, you could use those genes to define a gene set. So instead of it, so in this case, each of the bar can either be genes that are annotated as belonging to the same gene ontology code, or they could be genes from a previously published experiment, are all differentially expressed or upregulated in that experiment, or maybe downregulated in a different experiment. This way, when you are doing an experiment yourself, say you perturb the cell, and then you see some genes differentially expressed, you input the whole gene order into this experiment. And then maybe you go run through the GSEA, it will tell you not only maybe there are some biological functions or pathways that are enriched in your differentially expressed genes, it might also tell you that previously some people have done a similar experiment for differential expression, and those genes that are, for example, upregulated in breast cancer are in your differential gene list that are enriched, right? So in this case, you can use a GSEA to help find other published studies that might be related to yours. For example, if you're treating a cancer cell and you see some genes downregulated, and this group of genes happen to be also those genes that are causing a disease, then maybe that's an important drug you can use to treat the disease, right? That gives you a lot of hint whether your experiments, whatever drug treatment or disease, are relevant to some other published studies. Okay, so that's a very, very useful tool. So the summary here is gene ontology. We mentioned that it's a tree, it's an acyclic tree, and every gene is annotated to have 3 codes: molecular function, biological process, and cellular components, right? And there are many tools that are useful to help you annotate a group of genes that are enriched in any of these 3 things. And the one tool that we really recommend is this David tool. You will be using those in your homework, and you can see that it's pretty easy to use and give you some quite meaningful result. And then gene set enrichment is you don't really decide what group of genes to use. You just give it a whole gene order. And then you can use it to detect a general trend without setting the limit on where to cut off of differential genes. And it uses this Kamenov-Smirnov non-parametric test to see whether genes predefined are enriched in your upregulated genes or enriched in your downregulated genes. And here you do also need to do multiple hypothesis testing because you are testing many, many gene sets for either belonging to the same gene ontology group or belonging to another experiment which have shown to be differentially expressed. And since then, this molecular signature DB is part of the gene set enrichment to help define the gene sets, but also bring relevant studies to a user if you have your own differential expression. Yes, question?
I'm really sorry. Can you explain the KS test again, especially the plot?
Here?
Yeah.
Yeah, so basically you have 2 curves. One curve is to ask what percentage of genes have differential expression below a certain fold change, genome-wide of all the genes. So you can imagine very, very few genes will have a fold change of less than 0.1, which means after the whatever treatment or your differential condition, the treatment and control has a fold change. Basically, it means the treatment will decrease the gene X by tenfold. That is the fold change of 0.1. Then there will be a little bit more genes with the fold change below maybe like 0.5, and then there will be more fold change of below 1. And usually this one is maybe not the best example. Usually you would expect majority of those genes are not differentially expressed. So they should give you a fold change around 1. That's why this is not necessarily the best background. So supposedly this is the background. It should be around 50, where the fold change is 1, and about 50 of the genes are not differentially expressed. right? Their fold change will be close to 1. But then as you keep going, you ask what percentage of genes will have a fold change below 10? Well, almost none of the genes will have a fold change above 10. Therefore, 100% of the genes are below 10, right? That's your background distribution. Then you ask, for my gene set of interests, which is defined as sharing a particular gene ontology code, you ask how many percent of these genes have a fold change. So the y-axis is the percentage of genes in this group have a fold change below 0.1, below 0.5, below 1, below 10, and so on. Okay, and so this just asks, is the genes in this gene group very different from your genome-wide in terms of their cumulative differential expression level? And if they are very, very different, that's something interesting, right? You want to know this, even though individual gene may not really show you anything interesting. Okay. Is there another question behind? Yeah.
Oh, I was going to ask what the enrichment scores on the plots.
This enrichment score basically help you evaluate how skewed the gene sets are relative to genome-wide your distribution. You can see this is kind of like the D-value. What's the best separation between your genome-wide and your gene set of interest?
Does it matter what you actually remember when you look at these plots? You want like a certain shape, you want a certain
usually there is a lead enrichment score you can use, but most people use the p-value or the q-value. Yeah, it's a FDR adjusted p-value is the most indicative. Well, it gives you a rough idea how enriched your data is, but it depends on how big your list is and how skewed it is to the left or to the right. Okay, yeah, another question?
If you don't say something like that, that's what just tell you how far each other.
So KS test is a non-parametric test, right? Theoretically, if you know your distribution, you can use better statistics. For example, if microarray, you assume that it is a normal distribution after some whatever normalization, then you can use, for example, a t-test as well to look at, because you can see you are asking whether the group of genes you're interested in have a differential expression. You know, if you believe this is kind of a normal distribution, you can use a z-test as well. And so I would say probably over the years, gene set enrichment analysis have gradually also improved their analysis. I'm not sure that this algorithm has been optimized to look at negative binomial distribution from RNA-seq. By the way, a lot of the tools that we discussed in this class were initially developed for microarrays, and people were just continuing to use it for RNA-seq. They are probably decent, but may not really be the best. But a lot of people are now dealing with new type of bioinformatic problems now. They probably are not optimized for a negative binomial distribution. Okay, yeah, another question. Oh, GSEA is gene set enrichment analysis. Yeah. Is to look at, it's the basic idea is to ask whether the genes annotated as having some gene ontology or pathways are skewed to the left or to the right compared to, you know, all the genes. GWAS is to ask if you profile a lot of patients that are disease versus normal, to look at whether some genes are associated with the disease at the DNA level, not at the gene expression level. We will not have this course teaching you exactly how GWAS, and well, we will teach you roughly the basic idea of GWAS analysis, but we'll teach you how to interpret the GWAS analysis. That's mostly looking at, based on your genetic makeup, whether this person is likely to have a disease or not. Whereas in this case, we are looking at the cells in terms of expression, whether the normal versus the disease, maybe even from the same individual, are expressed in a different way. Okay, it's a very, very different type of tests. Okay.